Hey folks, welcome to the Density Stacking Intermolecular Forces Lab. So this one is really fun and beautiful. For this experiment, you're going to need all these materials here. You're going to need 2 of your test tubes in the test tube rack. You need food coloring. You're going to need some water. I just have a bottle of tap water here. You need some dish soap, some salt, some vegetable oil or corn oil or mineral oil, whatever oil. And 3 cups, Dixie cups are fine, just like little paper cups or plastic Solo cups. Those are actually best because then you can kind of pinch them and get a pour spout. Also like a measuring cup would work too, one that has a pour spout. And you need your 10 milliliter graduated cylinder, all right? And a spoon. Did I say food coloring? And a spoon or something to stir with. All right, so the first thing you need to do is you need to prepare your solutions. We're going to make some colored solutions. And so I guess, you'll also need your 100 milliliter graduated cylinder. So let me double check my procedure here. Yes, okay. So for the first one you're going to make is your salt water. This is going to be very concentrated salt water. So for this we want to pour water into our 100 milliliter graduated cylinder. We want exactly 100 milliliters, red at eye level to be exactly at the line. And I'm going to put that in this container here. And then the instructions say to add 20 grams of salt. So, you're also going to need your digital scale. Move some stuff aside here. You're going to need your digital scale and the weigh tray that comes with it. The lid is also a weigh tray. That's what we're going to use. And I'm going to turn on the scale, wait for it to say 0, and then I'm going to place the tray on here, and then I want to zero it out, so I want to hit the T button to tear it back to 0. And now I'm just going to pour until it gets to 20 grams. All right, it does not need to be exactly 20. This is not quantitative, it's qualitative, so close to 20. All right, I'm at 20.30 grams, that's fine. And we're going to pour that into our first cup and use a spoon to stir it up. All right, if the water is a little bit warm, and it's okay, I didn't get quite all of the salt off, but I'll wash that lid later. I'm just trying to make a very concentrated salt solution of salt water. Give that a good stir, all right? And we're also going to dye the solution because it's prettier this way. I suggest doing green for the salt water, unless your dish soap is green. So my dish soap is blue, so I'm going to make sure these are all different colors than my dish soap. So if your dish soap is green, make the salt water blue, all right? And it doesn't matter how much dye you add, this is just to visually enhance it, make it pretty. All right, so I've got some a green salt solution here. Get some white backing so you can see it better. I can put my scale away now. This nice, pretty green salt solution, and I want to stir it until all the salt is dissolved, till I can't see or feel or hear any gritty salt crystals. All right, we'll come back to that. In the second beaker, I'm going to just pour plain water. All right, 50 milliliters. It says to do 50 milliliters of plain water. It actually doesn't matter what volume, but I like to have you guys actually using the science equipment that is provided. You know, feel like a real scientist over there. And I suggest coloring the water, oops, not red, I said in the protocol, color it yellow, okay? Dye the water yellow. If your soap is yellow, dye it blue. All right, so I did a couple of drops, spilled, of yellow, and it's almost like kind of orangish. So maybe just one drop was all I needed to make it yellow, but again, doesn't matter, it's just making it pretty. And then I forgot another ingredient that you need, which is your isopropyl alcohol. But this one, your 70% isopropyl alcohol, we're going to add this to this container, 50 milliliters. You can measure it out using your graduated cylinder or just put some in a cup. We're just putting it in here to dye it, okay? And I say to dye the isopropanol red, but you guessed it, if your dish soap is red, then dye it blue, okay? And you can just kind of stir it with a spoon that you clean off from the salt solution or a separate spoon. So we've got these pretty colors. It looks like a traffic light. All right. So each of these liquids, before we dyed them, they were all the same color. They were all just transparent, clear. You wouldn't be able to tell them apart. So we added the food coloring just to give them a visual, make them visually different. Another thing that we can't see about them that is different is that they all have different densities. And when liquids have different densities, or things of different densities will stack on top of each other. And liquids, so anything that's like for water, if you put anything in water, if it's less dense than water, it will float. If it's more dense than water, it will sink. That's true of whether that thing is a solid or whether that thing is a liquid. The key, though, with liquids is to layer them gently so that they don't mix together. And so in this experiment, you do need to use the graduated cylinder for this next step. So now we can start stacking our things. So I've got these solutions, all of my solutions here, and then I've got my tubes. And I want to label them A and B. Let's do that. Maybe put a piece of tape on them or use a permanent marker and just label them A and B. This is a wax marker. All right, so we've got A and B. So the first thing we need to do is measure out. The instructions say we'll need to remove the screw caps from our tubes. Some didn't remove quite right. Whatever. First thing that we're going to do is add the salt solution. It's important to do this in the correct order, the order that it says to do it in the lab. So it says to add 6 milliliters of the green salt solution. So we're going to use our graduated cylinder, our 10 milliliter graduated cylinder, the little guy. Reading at eye level, 6 milliliters. Great. I'm going to add that to tube A. And I'm going to tilt the tube so that it's sliding down slowly and not plopping down. I don't want to hold the tube like this. All right, I want to hold it slanted. I'm going to do the same thing for tube B. 6 milliliters. Yep. And into tube B. Now, the next thing that we're supposed to add is, I'm going to put that salt solution aside. The next thing it tells us to add is the vegetable oil. Doing it in the correct order is important. So for the vegetable oil, it says only add 4 milliliters. So I'm going to measure out 4 milliliters. That's a little bit more than 4, so I'm going to dump some off. I have a little waste container. And there, that's perfect. I'm putting 4 milliliters in tube A. And again, I'm slanting it and pouring it very gently, very slowly down the side of the wall of the tube. And look how beautiful that is. All right, we're going to do this again for tube B. Perfect. Nailed it. 4 milliliters. Tilt your tube and pour it gently. Ta-da! Part of this lab is working on, just having good hands. And to be a scientist, you need to have good hands. You need to have good manual dexterity, be able to do this gently enough, right? The next thing on the list to add, I'm going to cap my oil and put that aside, is the plain water. The plain water was the one that I dyed yellow. And so I'm going to do 6 milliliters of this. Notice I'm not washing the graduated cylinder out in between steps because I am putting all of the same ingredients in each tube. So for this particular experiment, it's not necessary. Generally in chemistry, you would want to use clean devices. So you'd want to clean it out between each step. So I poured my yellow water in there. Oh, check that out. I don't have a career as a YouTuber. Ah, beautiful. All right, our layers. And notice they're not necessarily layering in the same order that we are adding them. So it's not necessary. We're not building this stacking on top, on top, on top. Some of these liquids are actually sinking below other liquids that we already have in there based on their density. So they're going to order themselves. They're going to layer in order of their densities. Oops. All right, so then solution number 4 that we're going to add to each of these is the isopropanol, and we dyed that one red, and it says to do 6 milliliters of that. So I'm measuring this out to 6 milliliters. I'm fudging it a little for this video because I don't want to be perfectionistic. That's boring to watch. But you should attempt to be perfectionistic. Try to get exactly 6. And voila. Et voila. All right. Now, the last ingredient is only going into tube B. Hold on, let me add isopropanol. I'm still tilting the tube, pouring it gently, slowly down the side wall of the container because I don't want to introduce, if I do it too forcefully, the liquids will mix, right? It'll break. It'll provide energy for the molecules to break apart and mix together. So I'm doing it as gently as possible. Now, we're going to, these currently look exactly the same. We have the exact same ingredients in them. And so let's just go ahead and cap tube A off. Tube B, we're going to add a 5th ingredient, and that is our dish soap. All right, for the dish soap, the instructions are to add 10 milliliters. All right, so I'm pouring it all the way up to the top line, 10 milliliters. All right, you might even want to add a little bit more than that. We need a good bit of soap. And we're going to add that again to our tube B. And dish soap is kind of thick, kind of viscous. You might not be able to get quite all of it out. That's also in part why we did a little bit more, a little higher volume. And ooh-hoo-hoo, check it out. All right, let it sit for a minute so they can level off. Oh, look what's happening. I don't know if you can see this. The blue and yellow are switching places. That dish soap is denser than the yellow solution, so it's falling and taking its rightful spot there between yellow and green. All right, so it's kind of cool to watch. So you'll want to cap both of these tubes and take a picture. Take a picture, a nice clear picture. Maybe you want to hold them like this. get your camera, snap a photo because you can't see it as well when they're sitting in the rack. The rack kind of gets in the way of the photo op. All right. And also use some crayons, markers, color pencils, what have you, in order to color the tube diagrams in the lab to represent your different density layers, and label it too. Make sure you tell me that green is what and blue is what, and tell me what each of those solutions are. It's important that I see that you know they're different things, and also your colors might not match up to mine if you didn't have blue dish soap. So yeah, all right. In part 2, so there's some questions that I ask you about, which is the most dense, which is the least dense, how do you know that? Use CER, make a claim, what's your evidence, and what's your reasoning? And also, I give you the molecular structures, the chemical structures of each of these different substances, these 5 different substances, and ask you to determine, based on their structure, are they polar, are they nonpolar, are they ionic, or are they amphipathic? And amphipathic means that it has a nonpolar region that's hydrophobic, it's afraid of water, and it has a polar or usually a charged region that is very hydrophilic, it loves water. And so they're kind of, I think of them as like ambidextrous molecules. You can be right-handed, or you can be left-handed, or you can be able to use both. Amphipathic, they can do both. They can be soluble in both polar and nonpolar. So right now, we can't tell which of these liquids are soluble in one another because we have carefully added them so that they stay separate based on their different densities. In part 2, after you answer the questions for part 1, you're going to do a little shake, 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 shake. All right, you're just going to shake them both up, and then you're going to let them settle for about 5 minutes. Right, you're going to get a little bit of foam from the soap. That is one of the side effects of soap, so we want to let that settle. And we want to see which of these solutions stay mixed, stay blended, and form a conglomerate solution, and which ones separate back out. And again, that is going to be due to that solubility rule of like dissolves like. So any of these substances that had similar intermolecular forces, that have similar polarity, if you will, so nonpolar and nonpolar, or polar and polar, polar and ionic can also go together, all right? Those are the rules of like dissolves like. So in our 2 tubes, they will have different volumes because you added 5 things to tube B and 4 things to tube A. All right, and we can already see some stuff kind of happening here. So tube A, it's only been about a minute, we're starting to see a sort of separation, and it looks like 2 layers. So there's 1 layer up here, and then the other 3 things seem to have mixed. But in tube B, it seems to be it's not settling out quite as much. There is this layer at the top of just foam. We're not counting that. We're just looking at the liquid layers. And there might be a little bit of a layer here. All right, but it's certainly smaller than that of the one in tube A. All right, and that's because, remember, in tube B, we added soap. And soap is a classic emulsifier. It's something we call a surfactant, and it has the ability to emulsify things. So that's a vocabulary word for you to know, so I won't share it in the video. But at the end of 5 minutes, maybe even at the end of 2 minutes, you might be able to see these distinct differences where one clearly separates out, and in the other, you might have some separation, but not a lot. I could play around with it. You could use a little bit more soap. The more soap you use, the better. We just might not have enough soap in here to fully emulsify the solution. So feel free when you do yours to use, just fill up this whole thing. Don't go to the 10 milliliters, go up, whatever, just fill it all the way. And you'll have a larger soap layer, and then you'll have maybe enough to fully emulsify and not have it separate out again. I don't know why I'm mixing it. That's not going to necessarily help. All right, so that is the density stacking rainbow lab. I like rainbows, if you can't tell. All right, enjoy.